Okay, hello everyone. My name is Qianchen and I am from NIOS lab, the Cite lab. Okay, my title for today's talk is aligning the percepts and concepts from a brief glance. This is one of the study of my PhD project. So if I were to show you an image for a very short time, what can you see? So like this. And over this kind of really brief presentation, all of us would agree that we cannot really see the details because we don't have enough time to scan to different areas of the image and inspect those details. So the question here is that, what did we actually experience at that brief glance? And based on previous research, they proposed some perspective contents of this brief moment of experience. The first type of content is that we experience a perceptual summary of the image. To understand what a perceptual summary is, we must go back to how the brain processes visual input. And there's this kind of mechanism called cortical magnification. And I'll explain what cortical magnification is. And basically, this is our visual field. This light blue circle in the very center is like our fixation. And these color rings surrounding it represents our peripheral visual field that gets further and further away from our fixation. Now, what happens is that our visual cortices devote a huge area to process this small area of input. So the visual input coming from our fixation gets processed by a lot of cortical resources, whereas these peripheral visual inputs get pulled and compressed into these smaller and smaller areas on the cortex. And the perceptual summary is a result of this compression where the brain computes the summary statistics of visual inputs from pulling information within each pulling region. And to understand what this kind of perceptual summary really looks like, there are algorithms that simulate this process. For example, the most famous one is Freeman and Simoncelli's visual Metamers model, published on Nature Neuroscience 2011. What they do is, if you put input a source image into this kind of image synthesis model, the model will overlay this kind of window over this image. And these window sizes mimic the pulling region of cortical magnification. And then it just synthesizes the pixels within each window to get a synthesized perceptual summary. And according to them, the synthesized summary differs from the original physically, but they look the same if you show both for a very brief glance. And we can actually quickly check if it actually looked the same. And I think you will, agree or disagree whether they actually look the same, but hold that thought for now because this is part of my study as well. Okay. All right, so that was perceptual summary. Aside from this perceptual summary, the previous research also suggests that we experience a conceptual summary of the image over a brief glance. And conceptual summary, aka gist, is the general semantic meaning of the natural scene, and it can often be expressed in a short sentence or few words. So for this image, for example, the gist of it would be woman eating strawberries from a bowl or something like that. So now we know that there are two components, perceptual and conceptual summary. The problem is that these kind of concepts are proposed by different people. And what's unknown is the relation between them. So for example, does perceptual summary explain conceptual summary fully? Just take these pair of images here. We already know that human faces are very salient in terms of perception. And we can imagine that in the form of conceptual summary, face would be one of the most first things to be kind of mentioned. but we can see that this synthesis here kind of scrambled the face by a lot. So here the question is, do these two images give us the same gist over a brief glance? And this is basically the question for my current study, which is to investigate the relation between perceptual and conceptual summaries and ask the question, do participants report the same gist to a natural scene image and its perceptual summary? To answer this question, we developed an online behavioral task in which we first present a natural scene image for a very short time and then mask it. And then we ask the participants to describe this image in 5 words, which is basically to collect the conceptual summary of the image. And this image itself can be either intact or synthesized to preserve the summary statistics. And in this task, we used 100 pairs of intact synthesized images, which makes 200 images in total. And we test the task on 100 online participants. And we allocate the stimuli in a way that for every pair of images, we tested on 40 participants, in which 20 was allocated to see the intact version, and then the other 20 was allocated to see the synthesized. So now that we finished the task, for every image pair, we'll get 100 words on the intact version, and then another 100 words from the synthesized version. And if we were to quickly visualize them in a word cloud, this is what they look like. These are real data. They kind of look similar, but kind of different as well. So the question is, how can we quantify the differences in these GIST reports? And we'll answer this question in 2 steps. First, we'll transform the words into numeric vectors so as to quantify the difference. And the second step is to compute and analyze the cosine similarity among vectors. And now I'll start with the first step. So let's say for this particular image, we have one of these participants, and this participant report, woman eat strawberry smile bowl to describe this image. Now, to vectorize these words, we'll make use of Global Common Crawl, a pre-trained set of word vectors containing 1.9 million vocabulary. And basically, each word in this set is kind of represented by a 300-dimension vector. And for word in this set, its vector is a numeric representation of its meaning relative to the other words in the set. And we just search and assign the corresponding vector to each word, and this is what we get. 5 vectors to represent 5 words. And to get the summary representation of the 5 words, we just average across the 5 vectors to get a 300-dimension mean vector to show as a summary representation of the GIST report of this participant to the image. And here onward, I'll refer to this kind of mean vector as a subject GIST vector. Okay, so for every image pair, we just repeat this vectorization process on each of 40 participants so as to get 20 subject GIST vector on the intact version representing the GIST report of 20 people, and then another 20 vectors representing the other 20 people. And here, the vectors can have, variance, but the variance can either come from individual differences in their tendency to, choose words and respond, and it can also come from the differences between images. So how can we quantify the unique differences in vectors introduced by image synthesis as independent from the response differences in each individual participant? And here comes our next step, which is to compute and analyze the cosine similarity among vectors. So cosine similarity is a kind of value that tells you how similar two non-zero vectors are. This kind of score ranges between -1 to 1, with the score closer to 1, meaning 2 vectors being more similar. And for each single image pair, we compute the cosine similarity score between every possible pair of subject gist vectors. So the result of this would be a 40 by 40 similarity matrix. So the x, y axis are both just the participants and their gist vectors. Most importantly, this kind of computation gives us 3 sets of unique similarity values depending on the subject gist vector pair types. So the first type is within intact similarity scores. And in this type, each score is computed from comparing a participant who sees the intact version of the image and another participant who also sees the intact version of the image. So that's why it's called within intact. Likewise, we also have within synthesized. And finally, we have intact versus synthesized, comparing a participant seeing the intact version of the image and the other participant seeing the synthesized version of the image. And for these 3 different sets of similarity scores, the similarity level would depend on different things. So for this within intact, because both, each score, both participants see the same image, the similarity level would depend solely on whether these 2 people have the same tendency to use certain words, basically the individual differences in their response. Likewise, for within synthesized, we also only have the individual differences. Finally, for intact synthesized, of course, because they're still 2 different people, they have their own individual tendencies, there are individual differences. But potentially, because they see the different versions of the same image, they can also be potentially differences in the responses induced by intact versus synthesized version of the image. So here, if the difference induced by image manipulation is not significant, then we expect 3 sets of similarity scores to represent individual differences only. And we expect all 3 sets of similarity values to come from the same distribution. In contrast, if the difference induced by image manipulation is significant, then these differences can drive intact synthesized value away, as in a different distribution from the other 2 sets. So to test this, we make use of Kolmogorov-Smirnov test, KS test, which tells you whether 2 sets of data comes from the same distribution. And we run 2 KS tests comparing intact synthesized with within intact and then within synthesized, and then obtain a p-values from each test. As an exploratory analysis, we set the alpha level to 0.05. And here comes the results. So for this particular image pair we've been seeing the whole time, we get a significant result for both KS tests. This means that for this particular image pair, the participants gave different gist to the intact and the synthesized images. And now I'll talk about, what about the rest of the 100 image pairs? So here I show the cumulative distribution of the p-value, KS test p-values of all 100 image pairs. The y-axis represents the cumulative count up to 100 image pairs, and then the x-axis is the p-value. And the dotted black line shows the level of p equals 0.05. And each color line represents the results of the KS tests. And importantly, we want to look at the intersection between this p 0.05 with the KS test results. And we can see that around 60 image pairs with significant results for intact, within intact versus intact synthesized, and another 60 image pairs with a significant result for within synthesized versus intact synthesized. What this means is that there are most of the image pairs have some level of gist differences between the in-tag and synthesized. And if we were to look at the image pairs that have a non-significant result for both tests, we only have 27 out of 100 image pairs. So in summary, in this experiment, we investigated how well the perceptual summary of an image explains the conceptual summary of it. And to that end, we collected and compared the conceptual summaries for the in-tag and synthesized images. And we want to look at whether they are just the same. And we, at least based on our own image set and our own data set, we found that perceptual summary basically cannot fully explain the conceptual summary. So the next question is, what is missing in the perceptual part that is captured by the gist report? So one possibility is that from an image, people's perception of that image is kind of much richer than what the perceptual summary shows, to an extent that people can actually see the difference between the in-tag and the synthesized. So this kind of rich perceptual contents were also kind of expressed in the gist, resulting in the gist difference. That's it. Thank you.
Thank you. Any question or comments? Nothing? No. It's hard to ask comments from me because I know about this, but
I have a comment. Or
yeah.
I thought it was a very interesting study, but I guess the difference between the conceptual level and the perceptual level is the relational information that the subject has to construct, which presumably would take more time, which would be beyond the, for example, if you perceive, for example, a circle and a triangle, you also, at the conceptual level, you're also thinking about the relationship between the circle and the triangle. Is it inside or outside? Is it above or below, and so forth? So are you really expecting that would, some people regard that as two different processes. So are you really expecting the perceptual level to be able to convey that information in such a short time?
So
there's a kind of a relational level at the relational information at the conceptual level that's different from the relational information that's at the perceptual level. I think there's a difference between, for example, having metric space or distance versus, say, having some more abstract concepts like above, below, or so forth. So I thought it was a fascinating study, but I was just trying to get an understanding of what you expect to be the relationship between the perceptual and the conceptual level.
The relationship between the perceptual and conceptual level.
Maybe Qianchen, can you go through the slide for the explaining paradigm with the timeline? Yeah, that one. So Steve, one of the things that you are mentioning is this: within 133 milliseconds, you expect that people wouldn't be able to extract the relational property or conceptual property. It's too fast.
Yeah. Some of I expect they'll be able to extract the words, but just having a collection of words is not the same as having a structure over those words. For example, say John loves Mary, for example, is not the same thing as Mary loves John, even though they contain the same words. John loves and Mary, that their relationship between the two had mean two different concepts. And so this very short time, or put it another way, the paradigm that you're doing would be very nice to distinguish between at what level do we extract the constituents of the scene and then put them together in a relational construction.
I see.
So yeah. So
I
yeah.
see. So, my understanding of your comments is that from this very short time, people may be able to spill out a few words, but then putting the words together in a coherent, in a particular way that expresses the meaning needs probably longer time. Is this?
Yeah, yeah. And presumably it's a back and forth between the perceptual level and the conceptual level. So the paradigm would be very nice if you could vary presumably you would get a different relationship between the perceptual level and the conceptual level when you change the response window, or sorry, the stimulus presentation time from 133 milliseconds to say up to 200 or 400 or 500 or something like that. So actually it'd be much more a dynamic.
That there are actually free report studies on this kind of typical paradigm, and they also show it for a very short time, and people can report coherent sentence or even write paragraphs, but the problem is that people may be very enthusiastic about these kind of things and then give really detailed descriptions in the first 3 trials, and then as time goes by, they just get tired and more tired, and they just spill just single words. And this 5 words kind of paradigm is actually based on Chu Ying's and other lab members kind of experiments, in a sense, like how many things we want them to report would maintain a kind of relatively optimal performance throughout. And 5 words seems to be the optimal. And that's why we use the 5 words to kind of extract the conceptual summary. But yeah, I agree. This kind of single words report can lose some kind of information, especially when we ask for single words.
Maybe I'll just also add a little bit to bridge the gap of the discussion, I think. So Steve, I think your expectation that with this short time window, people will be confused and they cannot report on the details and so on. That's totally absolutely correct. If you rely on that very traditional psychological literature. However, around towards the end of the 1996 or something like that, Simon Thorpe and other people have started to use natural scenes as a stimuli, very complex ones. And then we are kind of following that kind of tradition these days, but it's really counterintuitive in some way. But if you use very complex stimulus like a natural scene, then you can actually give a more complex, reports accurately.
Right.
The distinction between circular and triangle different orientation, that's much more difficult with 133 milliseconds. But the one shot of this complex thing is super easy to see for us.
Right.
And that's one of the the surprise. But we have been consistently thinking, seeing this throughout all the experiments. And one of the key is that its naturalness, that it's very fit to our brain. And also, we never repeat the stimulus, and that reduces confusion, unlike traditional psychology. Yeah.
Yeah, it seems a fascinating distinction between why people can do it so well in such apparently complex scenes, yet with more simpler cases, they seem to, very counterintuitive, which makes it very interesting to study, I think.
Thank you.